Hello everyone, and welcome to the nervous system movie. What you see here is a neuron, and we're going to talk about how they work in a second. First, we know the nervous system is very complex, so we divide it up so we can try to understand it. And the one major part is the central nervous system. The central nervous system, as you can see here, it consists of the brain. And then leaving the brain, we have the spinal cord. So the central nervous system consists of the brain and spinal cord, and its main function is information processing. Then the peripheral nervous system is everything outside of the central nervous system, and it can be divided into motor and sensory divisions. The sensory division sends information to the central nervous system, and the motor division is receiving information from the central nervous system. And the motor division can be further broken into somatic, which is our voluntary skeletal muscles, and then autonomic, which we'll talk about in chapter 14. Now there are three basic functions to the nervous system you should know. They are input, processing, and output. We can liken this to a computer. A computer gets input from its keyboard. That would be the input. And the processing happens in a processor somewhere down in that box. And then the output would be on the screen, or a printer would be a form of output for a computer. Your nervous system does these same three basic functions. And the basic structural and functional unit of the nervous system is the neuron. So we're going to look at a neuron structure here. Here you see the dendrites. They are highly branched processes. They receive input from other neurons, so they are the input region of the neuron. Then the cell body contains most of the organelles. Some integration happens here. Neurotransmitters are chemical messengers, and they're produced here in the cell body. Neurofilaments are the intermediate filaments in a neuron. They provide structure to all these branched processes, and they're also used to transport things down these long axons. So the axons, the next thing we're going to talk about, axons are the output region. The axon hillock is the base of the axon. That's where action potentials are first generated. The axolemma is simply the plasma membrane surrounding the axon. The axoplasm is the cytoplasm and the organelles it contains in the axon. And axons usually branch into what are called telodendria, which end at synaptic terminals, also sometimes called axon terminals. And where the synaptic terminal meets this next neuron, it forms a synapse. So here's a close-up on the synapse. This is where cells communicate, and they use neurotransmitters for this. The neurotransmitters are packaged in these green synaptic vesicles. And neurons can send messages to other neurons. As you see in this picture, other neurons, muscles, or glands. And many neurons have collateral branches, like you see here. Those collateral branches are going to allow this neuron to communicate with more than one other neurons. Most neurons lack centrioles, so they cannot divide. That's why once you have a spinal cord injury, we can't get it to regrow. But once your brain is formed, we don't want it to be changing. So this is a good thing. So most neurons have a lifespan of your whole life. So we have some review questions to help you understand. So the nervous system has 3 overlapping functions, and the logical order in which they occur is, well, first we have to have sensory input, then we have integration in the central nervous system, and then motor output. So number 3 is correct. While studying for an exam, you reach for a beverage. To extend your arm, your blank nervous system is activated. And since that's a voluntary muscle, that's your somatic nervous system. What part of a neuron is the output region? And the correct answer is axon. The portion of an axon that contacts its target is the axon terminal. Where are neurotransmitters made? And correct answer is cell body. What part of a neuron is the input region? And the correct answer is dendrite. What type of cells transmit action potentials? The correct answer to that is neurons. So we can classify neurons based on their structure or their function. So first we're going to look at the structural classification. This here is called an anaxonic neuron. These are very small. It's hard to tell what is an axon. This type of neuron is found only in the central nervous system, the CNS, and its job is information processing. The next structural class is bipolar. These have one dendrite and one axon, and their job is relaying information. They're found in the special senses. And this is a unipolar neuron. The dendrite and axon are continuous. The cell body is off to the side. And these are going to be sensory neurons. All your sensory neurons look like this, because they basically just relay the information from a receptor. And then finally we have the multipolar neurons. 2 or more dendrites and 1 axon. This is the most common type of neuron, and these are going to be motor neurons. And then functionally we can classify neurons as sensory neurons if they're bringing input to the central nervous system. We can classify them as interneurons if they're doing the processing. And we can classify them as motor neurons if they're doing the output, taking information away from the central nervous system. Alright, and this shows how everything works together. The sensory receptors are going to send information through the sensory division to the central nervous system for processing. It's going to go out, information goes out through the somatic and autonomic nervous system to the muscles. Or in the case of autonomic, it's smooth muscles, glands, cardiac muscle. So next we're going to talk about glial cells, sometimes called neuroglia. Their job is to support neurons. And we have different ones in the central and the peripheral nervous system. So first let's look at the central nervous system. This is from the spinal cord here. And these red cells here, they're not really red. For the purposes of this diagram, they're red. They're called ependymal cells. Their job is to produce cerebrospinal fluid, or CSF. We also have some specialized cells called ependymocytes, which circulate the cerebrospinal fluid. And they have cilia, you can see here. Tanocytes are important for transporting substances between the CSF and the brain. This type of glial cell here is called a microglia. They are the immune system of the central nervous system. So if any foreign invaders somehow got into the immune system, they would take care of it. And then we have astrocytes. Here are some astrocytes. As you can see here, they are forming what's called the blood-brain barrier. So they surround this blood vessel and keep bad things out of the central nervous system, hopefully. They also regulate ion, nutrient, and gas concentrations around neurons. They can absorb and recycle neurotransmitters. They can form scar tissue. They do a lot of stuff, so if you're ever not sure, guess astrocytes. Also in the CNS, we have oligodendrocytes. Here's oligodendrocytes. They form the myelin sheath, which you can see here. The myelin sheath insulates axons and speeds up action potentials. Myelinated axons up here white, and they're called white matter. And then unmyelinated axons, along with dendrites and cell bodies, appear gray and are called gray matter. In the peripheral nervous system, we have what are called Schwann cells. Schwann cells form the myelin sheath in the peripheral nervous system. And then as we can see in this picture, we have satellite cells. They surround cell bodies and regulate the environment, similar to the astrocytes in the central nervous system. Schwann cells are helpful in the repair of a damaged neuron. So if this neuron here gets damaged or cut somehow, what will happen is the axon and the myelin degenerate distal to the injury, so away from the injury. And then macrophages will come into cleaning the debris. Schwann cells will start to regrow along the original path of the axon. And you can see the macrophages cleaning up the debris. Then the axon, so the Schwann cells grow first, then the axon starts to regrow along the path established by the Schwann cells. Now this only happens in the peripheral nervous system. It does not happen in the central nervous system. And if you can find out why, you'll be very famous and very rich. Then step 4, new Schwann cells are going to surround this new axon and myelinate it. Alright, so review questions. What type of cells circulate cerebrospinal fluid? Anything to do with cerebrospinal fluid is going to be ependymal cells. What type of cells anchor neurons to their nutrient supplies? And that would be astrocytes. What part of a neuron is the output region? Correct answer is axon. Alright, so now we're going to talk about neurophysiology, which is how neurons work. And there are some different things that happen. First, resting potentials happen. Well, it has a resting potential at rest. It can generate graded potentials and action potentials, which then will lead to synaptic activity and information processing. But we're going to start talking about the membrane potential, or the transmembrane potential, which is the difference in charge across the cell membrane. So if we look at this as a typical cell, here's the inside of the cell, here's the outside of the cell, and we can see it's negative inside. It's due to these negatively charged proteins and differences in ionic concentrations. So there's more positively charged ions outside the cell than there are inside the cell. And to remember what those ionic concentrations are, remember this picture. Your neurons, or really all your cells, are like a banana in salt water. And bananas, First, the water, salt water. As people know, salt is sodium chloride, so there's high sodium outside the cell. And if you don't know, the chemical symbol for sodium is Na+, because sodium is an ion, a positively charged ion. And then inside the banana, most people know bananas are good sources of potassium, and the chemical symbol for potassium is K+. And it's a positively charged ion. And this resting potential, we're going to watch a movie. I'm going to go to Mastering A&P to find this movie. I'm going to go to the study area, and then go to A&P Flicks, and we're going to watch Resting Membrane Potential.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, so now we're going to do a review to see if anybody was paying attention. Value of the resting membrane potential is, correct answer is -70. Concentration gradients exist across the plasma membrane. Specifically, there are remember, it's a banana in salt water. So there's going to be more potassium inside and more sodium outside the plasma membrane. The plasma membrane is more permeable to potassium ions. Sodium-potassium pump transports sodium out and potassium into the cell. The resting membrane potential depends on each atom below its set. Concentration gradient for sodium ions. Yes. The greater permeability of the plasma membrane to potassium ions. Yes. And the greater number of potassium link channels, the greater permeability of the membrane to anions rather than cations is not correct. So C is the correct answer to that one. Okay. So this shows a channel. And our resting potential is due to these leak channels, which are always open. So here's a potassium and a sodium channel, and a cell has more potassium channels normally, that's why we'll get to that. So an undisturbed cell has potassium leaking out through leak channels, and then it has sodium leaking in through leak channels. They're just moving because of diffusion. And then you have the sodium-potassium pump working that's pumping sodium out and potassium back in to maintain the concentration gradients. And the ions move across the membrane because of their electrochemical gradient. The electrical gradient is due to charge attractions. Opposite charges attract and like charges repel. The chemical gradient is due to diffusion. Things always move from high concentration to low concentration. And the equilibrium potential is where the chemical and electrical forces are equal and opposite. So let's look at a hypothetical cell that's permeable only to potassium. So it has a chemical gradient. Potassium concentration is higher inside the cell, so due to its chemical gradient, it wants to leave the cell due to diffusion. But because potassium is positively charged, and the inside of the cell has more negative charges, the electrical gradient is pushing potassium into the cell. So the chemical and electrical gradient combined make up the electrochemical gradient, and combined it's going out of the cell. And if the cell was permeable only to potassium, potassium would leave the cell until the equilibrium potential was reached. And for potassium, that would be at about minus 90 millivolts. At that point, you would see the chemical gradient is not as strong, and the electrical gradient is getting stronger because there's more negative charges to attract positive sodium ions. So at that point, there would be no net movement of potassium. Now let's look at a cell that's permeable, another hypothetical cell permeable only to sodium. Sodium's chemical gradient is into the cell, because sodium has a higher concentration outside of the cell. Sodium's electrical gradient is also into the cell, because sodium is positively charged and the inside of the cell is negatively charged. So if the cell was permeable only to sodium, sodium would enter the cell until the equilibrium potential was reached. And for sodium, that would be about plus 66. So at that point, there's enough positive charges in here to counteract the chemical gradient. And a real cell at rest is much more permeable to potassium than K was. So the resting potential is close to potassium's equilibrium potential. Remember, the resting potential is about minus 70 for potassium's equilibrium potential that was minus 90. Alright, some more review questions. The voltage displayed across the resting neuron membrane is termed the resting membrane potential is correct. The resting plasma membrane is more permeable to, and the correct answer is potassium. Maintenance of the resting membrane potential is possible due to sodium ions passing through the plasma membrane. Yes, the action of the sodium potassium pump. Yes. Potassium ions spontaneously crossing to the interior of the neuron. That was not happening. So B is the correct answer. Sodium ions can't just pass through the plasma membrane, they have to go through an ion channel. The resting membrane potential depends on each item below except the concentration needed for sodium ions. Yes. Greater permeability of the plasma membrane to potassium ion, yes. And the greater number of potassium ion channels. So it does not depend on greater permeability of the membrane to anions rather than cations. The sodium potassium pump transports sodium out and potassium into the cell. Concentration gradients exist across the plasma membrane. Specifically, there are, but since it's a banana in salt water, there are more potassium inside and more sodium ions outside the plasma membrane. And the value of the resting membrane potential is -70. Alright, so before we talk about graded potentials, we need to talk about ion channels. Ion channels allow ions to cross the plasma membrane and change the transmembrane potential. Remember, normally ions cannot go through the plasma membrane because it's a lipid bilayer. And what we see here is called a chemically gated channel. Chemically gated channels open with a chemical messenger or a neurotransmitter. In this case, it's acetylcholine. When a neurotransmitter binds to them, they open. When the neurotransmitter is bound, as we see here, the channel is open. And when no neurotransmitter is bound, the channel is closed. Then we also have voltage-gated channels, which open or close in response to changes in voltage or charge. You can see at -70, this channel is closed. At -60, it opens. So when the inside of the cell becomes less negative, the channel opens and ions can flow across. And the way a voltage-gated channel works, remember these are proteins, so they're made up of amino acids. And some amino acids are positively charged, and some are negatively charged. This region of the protein right here has a lot of positively charged amino acids. So when the inside of the cell is negative, they attract this positive charge and keep this channel closed. When some positive ions start to come in, and their positive charges will repel the positive charges in this part of the protein and open this channel. So that's how we think voltage-gated channels work. And there's one more type of channel, the mechanically gated channels, and they open in response to physical disturbance in the plasma membrane. So this is how touch receptors work. It's through these mechanically gated channels. Okay, next we are going to talk about graded potentials. So graded potentials are short-lived and they are local changes in transmembrane potential. So at rest, a cell's chemically gated sodium channels are closed. As you can see here, the channels are closed, no ions are flowing. When a neurotransmitter binds to a chemically gated sodium channel, as you see here, it opens and positive sodium ions enter, and then these sodium ions, they spread out and generate a local current. And as you can see, the amount of depolarization, or the change in charge, here right at the stimulation site it's minus 60, and as you get farther away it's not as much of a change, and when you get really far away the change is 0. So the degree of depolarization decreases with distance away from the stimulation site. So that's why we say graded potentials are local. They only affect the area right around them. And this figure shows that as well. This is showing the amount of depolarization, and closer to the stimulation site it's higher, and as you get farther away it's less and less. And the change in the transmembrane potential also depends on the size of the stimulus. So a greater stimulus will lead to more channels opening. That will lead to more sodium ions entering. So graded potentials can be different sizes. And when a chemical opens sodium channels, as we see here, positive sodium ions enter the cell and the transmembrane potential becomes less negative. This is called a depolarization, or an EPSP, which stands for excitatory postsynaptic potential. When a chemical opens potassium channels, positive ions are going to leave the cell, going down their concentration gradient, their electrochemical gradient. And the cell becomes more negative. This is called a hyperpolarization, or an IPSP, which stands for inhibitory postsynaptic potential. So to review, graded potentials are short-lived, local, passive, meaning they don't require any energy, and they're variable in size depending on the strength of stimulation. So graded potentials, this is a question. Graded potentials, they're not long-lasting, they don't travel long distances, they're not self-generating, so none of these is the answer. Graded potentials can be depolarizing or hyperpolarizing, that's true. They are not caused by the opening of voltage-gated channels, they are caused by the opening of chemically gated channels, so A and C is correct for this one. So next we're going to talk about action potentials, and we're going to start with a movie. So we're going to go back to AMP Flicks and watch the generation of an action potential now.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary. Generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Okay, so the action potential is initiated in the correct answer is axon hillock, and the threshold stimulus immediately. The correct answer is B. It opens voltage-gated sodium channels. The membrane potential returns to the resting state due to the diffusion of ions through leak channels. And here's another way of looking at the generation of an action potential. I've kind of simplified it. So here's your neurotransmitter acetylcholine. It's going to bind to a chemically gated channel right here. That's going to open, allow sodium to flow in, down its electrochemical gradient. And some of that sodium is going to diffuse down here. Now it's going to open a voltage-gated sodium channel. If it's positive, so it open that voltage-gated channel, and more sodium enters. That sodium diffuses down here. Meanwhile, acetylcholine diffuses away, and this channel closes. This channel opens. More sodium enters. This channel back here closes. This sodium opens this voltage-gated channel, and even more sodium enters. And that continues all the way down the cell. So let's look at this step by step. So in a resting cell, all of the voltage-gated channels are closed, as we see here. If a graded potential from the opening of chemical gated channels is strong enough, it will bring a cell to what's called threshold, and that's the point where the voltage-gated sodium channels are going to open. And as the voltage-gated sodium channels open, more positive sodium ions enter the cell, raising the transmembrane potential, and this is known as depolarization. So there's more depolarization. When the cell reaches about plus 30, the voltage-gated sodium channels inactivate, and voltage-gated potassium channels open. Now this is going to allow potassium to leave the cell, and this is known as repolarization. Then this cell membrane is going to go down to about minus 90. And this phase is known as hyperpolarization because the potassium channels take a long time to close. And then finally all the voltage-gated channels close and the cell returns to the resting state. So here's the summary of the whole process. At rest, all the voltage-gated channels are closed. A chemical gated channel is going to open. That's going to bring the cell to threshold. At threshold, the voltage-gated channels start to open. more sodium enters, the cell becomes more positive, then those voltage-gated channels are going to inactivate, and the voltage-gated potassium channels open, and the cell hyperpolarizes, then more negative than the resting membrane potential, and then each of the channels goes back to the resting potential. And this is a graph of an action potential. So here we are at rest. Something opens a chemically gated channel, and that allows some positive ions to come in and make the cell more positive. When it reaches threshold, the voltage-gated sodium channels are going to open. Lots of sodium channels are going to rush in. That's depolarization. Then the sodium channels close and the voltage-gated potassium channels open. As positive potassium ions leave, that brings the cell, makes the cell more negative. That's repolarization. And because it takes those potassium channels a long time to close, it goes even lower than the resting potential. That's known as hyperpolarization. And then it will return to the resting potential. During the absolute refractory period, you see right here, this is the time when another action potential cannot be generated, because you have to open sodium channels to generate an action potential. If they're already open, they can't open. And during this period here, they're what's called inactivated. Then you also have a relative refractory period. This is when it takes a much larger than normal stimulus to generate an action potential. Threshold is the point where an action potential happens. So if you reach this point, you get an action potential. Even if that EPSP brought you up to here, up to minus 40 or minus 60, you still get the same size action potential. Action potentials are always the same size. We have what's called the all or none principle. It states that an action potential either happens or not. So no matter how big the graded potential is, as long as it brings the cell to threshold, you'll get an action potential. Alright, so this is going to show the process with more details. Again, inside the cell, outside the cell, acetylcholine is going to come in and bind to a chemically gated sodium channel. That's going to allow sodium to come in down its electrochemical gradient. That sodium is going to diffuse down and open a voltage-gated sodium channel. More sodium is going to enter, and it's going to diffuse down. Meanwhile, acetylcholine goes away. This channel closes. And this potassium channel is going to open, so this part of the membrane is now in repolarization, and over here it's going to be depolarization. So the sodium is coming in, depolarizing this part of the cell, while this sodium channel closes and this potassium channel opens, so this part of the membrane is now in repolarization. And then over here we're having depolarization as sodium enters. Potassium is going to leave, at least potassium channels are going to be open again. We have a potassium channel opening, and we've got the repolarization happening there, and that continues all the way down the neuron, and then the sodium potassium pump works to pump sodium out and potassium back into the cell to get the concentration back to the resting potential. Alright, action potentials are self-propagating, and they can travel long distances, and there is a nice movie about this we're going to watch on Mastering AMP.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Okay. The influx of sodium ions in one segment of an unmyelinated axon initiates which of the following events? And it will bring an action potential in that segment and a depolarizing current that brings the adjacent segment to threshold. It does not bring a repolarizing current. So A and B are correct. Action potential propagation in myelinated axons is known as, and the correct answer is E, saltatory conduction. In myelinated axons, action potential occurs at the nodes of Ranvier, and they propagate rapidly, so D is correct. The refractory period occurs when the voltage-gated sodium channels are inactivated and potassium channels are open. Alright, so let's review the steps of action potential propagation. An action potential is generated at one site, so here's where it's generated. Sodium ions enter the cell. These sodium ions bring the adjacent membrane over here to threshold. So that's what's happening here. They spread out and they bring this area to threshold. This opens more voltage-gated sodium channels, and the action potential travels in one direction because of the refractory period. And so then this is in refractory period, so the action potential can only travel this way. So the action potential moves down this way, and then as sodium ions enter here, they're going to spread out and bring this area here to threshold, and it's going to continue to spread like this all the way down the neuron. And if we look at a myelinated axon, such as this, in this myelinated segment, the action potential doesn't have to be generated there. It just kind of jumps from node to node. So propagation in a myelinated axon is faster because it only needs to be regenerated at the nodes. The current travels from node to node, and the action potential is regenerated at each node. So it's generated here, then here, and eventually will be generated here. That's how it's able to travel faster. And the velocity of action potentials depends on the degree of myelination and the axon diameter. A bigger diameter is going to give you a faster action potential because there's less resistance. Alright, next topic is the synapse. At the synapse, information travels from the presynaptic cell to the postsynaptic cell. The neuromuscular junction is a specialized synapse between nerve and a muscle, and we looked at that in Chapter 9. So let's look at the steps that occur at the synapse. So this is the presynaptic neuron here, and this is the postsynaptic neuron. An action potential comes, that's what this red thing is, the action potential. It's going to depolarize the synaptic knob, or the axon terminal, whatever you want to call this. This is going to open voltage-gated calcium channels, and calcium ions enter the cell. Then calcium binds to proteins on the synaptic vesicles, and this is what a synaptic vesicle looks like up close. It has several different proteins on it. Some, like this, are involved in packaging the neurotransmitters inside it. Some of these have calcium binding sites. Some of these help this bind with the plasma membrane of the neuron. So synaptotagmin and synaptobrevin are some that have calcium binding sites. And so when the calcium binds to them, it causes these synaptic vesicles to fuse with the plasma membrane, releasing its contents, as we see here. So if we look up close, what happens is the proteins in the synaptic vesicle zip together with these proteins on the plasma membrane, and that causes this fusion that you see here, and they release their contents. And the neurotransmitters, they are going to fuse across the synaptic cleft, and so this leads to release of neurotransmitters from the presynaptic cell. They diffuse across the synapse and bind to receptors. This will cause a gradient potential and possibly an action potential. If that gradient potential is big enough to bring this to threshold, you'll get an action potential in the postsynaptic cell. But this effect doesn't last long because the neurotransmitters are broken down and they diffuse away. So here's another view. Action potential depolarizes the synaptic knob, opens voltage-gated calcium channels. Calcium binds to the synaptic vesicles, allows them to fuse with the plasma membrane. They release their contents, which then is going to bind to receptors on the postsynaptic membrane. That's going to cause a gradient potential and possibly an action potential on the postsynaptic cell. And sometimes we have electrical synapses in the CNS. These are faster because The cells are connected by gap junctions, and an action potential can just travel from one neuron to the next. There's no need for neurotransmitters. But electrical synapses are not as versatile. They can only do one thing. Alright, we're going to talk a little bit about information processing and postsynaptic potentials. So EPSPs and IPSPs are the postsynaptic potentials. They are generated in the postsynaptic membrane. A single neuron may receive input from thousands of other neurons. So here's one neuron, and all these purple neurons and yellow ones are different neurons that are sending its signals. And in reality, it could be thousands. And the input from all of these different neurons is integrated in the cell body. And if we look at a single EPSP, it's probably not going to be able to generate an action potential. EPSPs and IPSPs, they can combine by what's called temporal or spatial summation. So in temporal summation, this is one axon terminal here, and if two stimuli come close enough in time, they can add together, and so one by itself would not be enough to bring this axon, this neuron to threshold, but two together close in time can combine to generate an action potential. With spatial summation, which is more common, you would have two different axon terminals touching this neuron, and if they're stimulated at the same time, they're able to bring this neuron to threshold, whereas if it was just one of them or the other, they would not be able to bring it to threshold. Information processing involves interacting groups of neurons, and information is encoded in the frequency and the pattern of action potentials. And the nervous system is very complex because we have regulatory neurons that can affect the activities of the presynaptic and the postsynaptic cells. Neurons can use over 100 different types of neurotransmitters. And they have different receptors, and depending on the stimulus, a neuron may release different or more than one neurotransmitter, which can have different effects. So it's very, very complicated. Here are some examples of neurotransmitters. We can classify them chemically. We're not going to worry about that for this class. We can classify them functionally as excitatory or inhibitory. That can also depend on the receptor. And depending on the receptor, a neurotransmitter can have immediate effects or slower, longer-lasting effects through second messengers. And this shows the frequency of action potentials is another way data can be stored or communicated. This is what we saw with muscles. A low frequency of action potentials gives you a small contraction. A higher frequency would give you more tension. Same thing with light touch. A light touch would generate a low frequency of action potentials, where a heavier touch or a harder touch or a heavier weight would give a higher frequency of action potentials.